Welcome to Heartline Biology, reminding you that biology has its own language, and we love to name everything in biology. Today we're focused on DNA replication. What is DNA replication? Whenever living things grow and get bigger, their cells are dividing. Cell division is an essential process for life, not only for living things to grow and get bigger, but also for living things to reproduce. For example, bacteria, we start off with one cell and they divide, and before you know it, we have a huge population of bacteria. Cell division is essential for life. So back to DNA replication, what is it? It's a process where DNA is copied in order for cells to divide successfully. Notice this cell starts off with that one little circle in the center, and then there's two. That circle in the center represents the DNA. It must copy itself for each daughter cell to get a copy of the DNA at the end of cell division. DNA replication is a process where DNA is copied in order to produce identical DNA molecules. And this must happen before cell division. Otherwise, cell division won't be successful. Each daughter cell needs to have an identical copy of DNA. Let's look at a hypothetical scenario where this cell is going to divide without DNA replication. The cell gets bigger, that one DNA molecule moves into one daughter cell, and the other daughter cell has no DNA. The result, that cell without the DNA wouldn't survive very long and would die. Therefore, cell division was not successful. On the other hand, when DNA is replicated, you end up with two copies of the DNA. One copy of DNA goes into each cell, and now you have two cells where both will survive. What's happening during DNA replication? To answer this, we need to look a little bit at the structure of DNA, beginning with its building blocks called nucleotides. Nucleotides have three segments: a sugar, a phosphate group, and a base. And that base can be an A, a T, a G, or a C. Let's look at these nucleotides and how we can string them together to form a single strand of DNA. There you can see a sequence of nucleotides bonded to each other, forming single-stranded DNA. But DNA is not single-stranded, it's double-stranded. There we go, there's the second strand of DNA. A couple things that's important to recognize about the structure of DNA here before we jump into DNA replication. Firstly, DNA has complementary base pairing, meaning that wherever there's an A on one strand, it will pair with a T on the other strand. Wherever there's a G on one strand, it pairs with a C on the opposite strand. Again, we call this complementary base pairing. The other thing that's important to recognize is that DNA has directionality, where the direction of the molecule is different. And this comes back to the nucleotide. On the sugar of the nucleotide, there's 5 carbons, and scientists number them 1, 2, 3, 4, and 5 prime. We use the 3 prime and the 5 prime to show the direction of the DNA molecule. The 3 prime is on the bottom of the sugar, and the 5 prime is towards the top of the sugar near the phosphate group. So let's look at the directionality of these DNA molecules. Every DNA strand has a 5 prime end and a 3 prime end. The bottom strand, we can see that the phosphate group is poking out on this strand on this side, and that's the 5 prime end. Whereas on the other side, it's the bottom of the sugar, and that tells us it's the 3 prime end. Looking at the top strand, we see the phosphate group poking out over here, and that is going to be the 5 prime end. And then on the other side of that strand, we see the bottom of the sugar. That tells us it's the 3 prime end. We call this directionality and the orientation of DNA anti-parallel, meaning opposites, because one strand goes 3 prime to 5 prime, the other one goes 5 prime to 3 prime. Parallel, because the two strands are side by side with each other and they do not cross each other, like railroad tracks. Anti-parallel just describes the feature of DNA molecules that the DNA strands are parallel, but they have opposite directionality. Now let's dive into what's happening in DNA replication. first thing that happens is that the DNA strands must separate from each other. Let's do that now. The next thing that happens is that new DNA is built to complement to the existing DNA strands. Right, notice at the bottom of the bottom strand, we have a G. In order to pair that successfully, it needs to be paired with a C, right? Well, there we go. And then we add on one nucleotide at a time, pairing to the existing strand using complementary base pairing, growing the DNA molecule one nucleotide at a time. This happens in a way that the DNA is built from 5' to 3'. That is, on the new strand, it's built from the 5' end, adding on to the 3' end of the growing new molecule. This is consistent for all DNA molecules. DNA is built from 5' to 3'. So we built the bottom strand, now that's double-stranded. The top strand will also get complemented to form identical double-stranded DNA here too. This, in a nutshell, is what's happening during DNA replication.